Welcome to Level 3, and this is Lecture 24, the opening lecture of Microbiology Fundamentals. For the last 11 lectures, we lived in the world of documents, data, and chemistry. Numbers that sit still until you measure them. Starting today, that changes. We are moving into the world of living organisms, and living organisms behave completely differently from any hazard you have dealt with so far in this series. Most quality professionals think of microbiology as the other lab down the hall. Something the chemists do not need to fully understand, and something microbiologists learn mostly on the job. I want to change that starting today. This lecture explains why microbiology sits at the center of product safety, and why getting it wrong costs more than any chemistry deviation ever will. Here is what we will cover today. Number 1. You will understand why microbiology demands a different mindset than chemical QC. A chemical residue does not multiply. A microorganism does. That single fact changes how you sample, how you test, and how you interpret every result you will ever see. Number 2, you will see how microbial contamination causes real recalls, real illness, and real death. Not just a citation in an inspection report, but actual harm to actual people. Number 3, you will know where contamination actually comes from. Raw materials, water, personnel, environment, and equipment. 5 sources that come up again and again in every investigation you will ever run. Number 4, you will understand the regulatory framework that governs microbiology. FDA, EU GMP Annex 1, ISO 22000, Codex HACCP, and the relevant USP chapters. It is a denser web of rules than what we covered in Level 2, and you need to know which rule applies where. Number 5, you will get a preview of the toolkit and the roadmap for the next 11 lectures. From gram staining, all the way to Salmonella PCR. Let me start with the single most important mental shift you need to make. In chemical QC, a contaminant sits still. A residue at 2 in the morning is the same residue at 8 in the morning. If you retest that sample tomorrow, you get essentially the same answer, within the precision of your method. Root cause almost always comes down to an instrument, a method, or an analyst error. And you usually have your result within hours. In microbiology, none of that is true. A colony count at 2 in the morning can be 10 times higher by 8 in the morning, because the organisms are actively multiplying. Your result depends heavily on incubation time, temperature, and growth conditions. If you retest the same sample after 48 hours, you are not testing the same population anymore, you are testing a population that has already grown, died back, or shifted in composition. Root cause is not just an equipment problem, it is a contamination source, plus the conditions that allowed it to grow. And results typically take days. For some organisms, up to 2 weeks. If you carry only one idea out of this slide, carry this one. Microbiology is not chemistry with more steps. It is a fundamentally different kind of science, and it demands its own rules. So why does this actually matter? Let me put some scale behind it. According to the World Health Organization, an estimated 866 million people fall ill every year from unsafe food, worldwide. And 1.5 million of them die. 29% of that health burden falls on children under 5. And globally, unsafe food costs an estimated $310 billion a year in lost productivity and medical expenses. These are not abstract numbers from a textbook. This is happening constantly, all over the world, and it traces back to exactly the kind of contamination we are about to spend the next 11 lectures learning to detect, prevent, and investigate. And if you work in pharmaceuticals rather than food, do not tune out here. The stakes are the same in kind. Sterility failures, objectionable organisms found in non-sterile products, contaminated water systems. These have caused facility shutdowns, product recalls, and patient harm. And very often, the root cause traces back to a single missed environmental monitoring trend that nobody acted on in time. Let me make this concrete with a real case, because numbers on a slide do not hit the same way a real event does. In 2011, whole cantaloupes grown at a single farm in Colorado caused the deadliest foodborne outbreak in the United States in decades. 147 people got sick. 33 people died. The outbreak reached 28 states. Here is what actually happened. The farm had recently switched to used processing equipment that had previously been used for washing potatoes. They skipped an antimicrobial wash step that should have been part of the process. And critically, they did not pre-cool the melons before putting them into cold storage. That last point matters enormously, because it meant condensation formed on the fruit once it went into the cooler. And Listeria monocytogenes does something most people do not expect: it keeps growing at refrigeration temperatures. So that condensation created exactly the moist, cool environment Listeria needed to multiply on the rind and on the processing equipment itself. Here is the detail that should genuinely unsettle you. This farm had passed a third-party food safety audit just days before the outbreak began. The lesson is not subtle. A passing audit is not proof of control. Equipment design and cleaning validation matter every bit as much as any laboratory result you will ever produce. I want you to hold on to that image of a passing audit sitting right next to an active outbreak, because you will see versions of that gap again throughout this level. Take a look at the photo on the right. That is the real scale of what a recall like this looks like once it reaches the public. The actual product, pulled from shelves, with a nation's worth of grocery stores affected. Sitting that image next to the numbers we just went through makes this case stick with you far more than description alone ever could. Now let us get systematic. Where does contamination actually come from? In almost every investigation you will run in this field, the source traces back to one of 5 categories: raw materials. Agricultural inputs carry native microflora from the field. Soil, animal contact, irrigation water. These are common entry points before processing has even started. Water systems. This is one of the highest risk vectors in any facility. Process water and rinse water can carry contamination directly onto product, and biofilm inside pipework can seed a facility with a resident contamination problem for years if it is not properly monitored. Personnel. Hands, hair, and respiratory droplets. Gowning breaches and poor hand hygiene are consistently cited in contamination investigations, across both food and pharmaceutical settings. Environment. Air handling systems, floor drains, and any surface prone to condensation. Listeria in particular favors exactly these damp, cool environmental niches, which is exactly what happened in the case we just discussed. And equipment. Hard to clean surfaces, dead legs in piping, and reused equipment brought in from another process. This is exactly what caused the case on the previous slide, and it is worth remembering because it comes up constantly. You do not need to memorize a textbook definition of any of these. You need to be able to walk into a facility and immediately think through all 5. Every single time. Now, the regulatory side. This is denser than what we covered in Level 2, so let me walk through it carefully. FDA 21 CFR Parts 110 and 117 govern U.S. food facilities. This is current good manufacturing practice and preventive controls for human food, including sanitation and pest control requirements. FDA 21 CFR Part 211 governs U.S. pharmaceuticals. This is the CGMP regulation for finished pharmaceuticals, and it covers microbial limits, environmental monitoring, and water systems. EU GMP Annex 1 governs the manufacture of sterile medicinal products in the EU. It covers contamination control strategy and cleanroom classification, and it was significantly revised in recent years to raise expectations around contamination control. ISO 22000 and Codex HACCP apply globally across the food supply chain. These are food safety management systems built around hazard analysis and critical control points, the same HACCP framework you may already have encountered if you work in food. USP Chapters 61, 62, and 71 govern U.S. Pharmacobial testing. These cover microbial enumeration, testing for objectionable organisms, and sterility test methods specifically. And ISO IEC 17025 sets general competence requirements for calibration and testing laboratories, including microbiology labs specifically. You do not need to memorize every number on this slide today. But you do need to know that this web of regulation exists, and you need to know roughly which framework applies to which part of your work. Because inspectors will expect you to know it. Here is a framework that will save you a lot of confusion for the rest of this level. Almost every method we cover from here forward exists to answer one of exactly two questions. The first question is, how many? This is quantitative testing. It counts the total living organisms in a sample, regardless of species, and it answers whether the overall bioburden is within an acceptable range for that product. Later in this level, this is what total plate count in Lecture 28 and yeast and mold count in Lecture 29 are doing. The result is always a number, colony-forming units per gram or per milliliter, compared against a numeric limit. The second question is, which ones? This is qualitative or pathogen testing. It detects the presence or absence of a specific, named organism, regardless of how many are present. It answers whether a dangerous species is present at all. This is what Salmonella testing in Lecture 31, Listeria monocytogenes testing in Lecture 32, and Staphylococcus aureus testing in Lecture 33 are all doing. The result here is presence or absence, and in most specifications, the tolerance for a positive result is 0. I want to be direct about why this slide matters. Confusing these two questions, or applying the wrong specification to the wrong question, is one of the most common errors new QC microbiologists make. A passing total plate count tells you nothing about whether Salmonella is present. Keep these two questions separate in your head from this point forward. Now, something a little different. From this point in the series onward, I want you looking at real pictures of real organisms, not just descriptions. You will see 6 organisms on this slide, each with a real photo next to it. Take a moment on each one before moving on. Start with Salmonella species. Notice the rod shape and the pink to red coloring that is characteristic of this genus once it is stained or grown out. Now Listeria monocytogenes. Notice the short rods, and if you are looking at a colony, how it presents on Oxford agar. Now E. coli 0157H7. Notice the rod shape, or, if you are looking at a colony image, how it appears on selective media like MacConkey or CHRO-Magar. Now Staphylococcus aureus. Notice the grape-like clusters that give this organism its name, or the gold coloring if the photo shows a colony on mannitol salt agar. Now Aspergillus, a mold. Notice the green or black fuzzy texture. Once you have seen that texture, you will never mistake it for anything else again. And finally, a yeast colony. Notice how cream-colored and smooth it looks, close enough to a bacterial colony that it is genuinely easy to misidentify early in your career. This is not busywork. When you see an unusual colony on a plate 6 months from now, I want your brain to already have a visual reference to compare it against. Reading a description is not the same as having seen the real thing. And now you have. Let us preview the toolkit you will be using throughout this level. 2 families of methods run through everything we cover, and you will often use both on the very same sample. The first family is classical, culture-based testing. This means growing organisms on selective agar and counting or identifying the resulting colonies. It is slower, anywhere from 24 hours to 14 days depending on the organism, but it remains the regulatory gold standard, and in most cases you cannot skip it even if you also run a rapid method. The second family is rapid, molecular testing. PCR, ADP bioluminescence, and immunoassays detect organisms, or markers left behind by organisms, in hours rather than days. These are typically used for screening and for faster release decisions, but they usually complement the classical methods rather than replacing them entirely. A technician streaking a plate inside a laminar flow hood. A real-time PCR thermocycler. And an ADP swab with its handheld luminometer reader. These are the tools you will be living with for the rest of this level, so get familiar with what they actually look like. Before we close, let me flag the mistakes I see most often from people who are new to microbiology QC. If you can avoid these 5 from day 1, you will be ahead of most of your peers. The first mistake is treating a high count as just retest it. A rising trend across repeated retests can itself be a signal of a real contamination event. Investigate the pattern, not just whichever number happens to come back last. The second mistake is ignoring incubation time and temperature. The wrong conditions will under-recover or over-recover organisms. Every validated method has a specific time and temperature. Follow it exactly every time. The third mistake is confusing total counts with pathogen testing, which we already covered on slide 8, but it is worth repeating here because it is genuinely the most common error. A passing total plate count says absolutely nothing about whether Salmonella or Listeria is present. The fourth mistake is underestimating sampling technique. Poor swabbing or poor plating technique introduces more variability into your results than almost any instrument ever will. And the fifth mistake is delaying the review of environmental monitoring trends. A single excursion is just one data point, but a trend building across several weeks is very often the earliest warning you will get of a real contamination source, before it ever shows up in a finished product result. Let us bring this lecture to a close with the key takeaways. Number 1. Microorganisms multiply. Chemical residues do not. That is the entire reason microbiology needs its own sampling, timing, and interpretation rules, separate from anything you learned in Level 2. Number 2. Unsafe food sickens an estimated 866 million people and kills 1.5 million people every year, worldwide. This is not a theoretical risk sitting in a textbook. It is happening right now. Number 3. Microbiology sits inside a dense regulatory web. FDA, EU GMP Annex 1, ISO 22000 Codex HACCP, the relevant USP chapters, and ISO 17025. Know which one applies to your work. Number 4. Every method you will learn in this level answers one of exactly 2 questions. How many organisms are present, which is quantitative? Or is a specific dangerous species present, which is qualitative? Number 5. Contamination has 5 recurring sources: raw materials, water, personnel, environment, and equipment. Know all 5 before you ever start an investigation. Number 6. This level runs from Lecture 24 through Lecture 35, moving from these fundamentals into gram staining, plate counts, and named pathogens like Salmonella and Listeria specifically. That is Lecture 24 complete. And with it, Level 3 has officially begun. You now understand why microbiology is treated so differently from chemistry in a QC environment, where contamination actually comes from, the regulatory framework that governs it, and the 2 core questions that every method in this level is built to answer. Up next is Lecture 25, Bacteria, Fungi, and Viruses. The basics every QC analyst needs. That is where we start building the biology itself, the foundation everything else in this level rests on. Take a look at the roadmap on this slide. 12 lectures, from where we are right now all the way through to anaerobic and microaerophilic organisms in Lecture 35. Every one of those builds directly on what we covered today, so if anything here felt new, it is worth sitting with before you move on. If this lecture helped you, please give it a like. Subscribe for new lectures every week. And drop your questions in the comments. I read every single one and I reply. I will see you in Lecture 25.